It should not be happening. First-dose anaphylaxis to a monoclonal antibody is rare, less than 1% of patients. But at hospitals across the American South, the number is closer to 20. Patients are crashing on their very first infusion of cetuximab, a newly approved cancer drug. Some are dying. The oncologists are baffled. They call in the allergists. This is the story of a drug that didn't just treat cancer. It revealed a hidden secret of human evolution. Thomas Platts Mills has been an allergist and immunologist at UVA for decades. He has seen unusual presentations before, but this one, first-dose reactions to a drug patients have literally never received, is extraordinary. It means the immune system was primed before the drug arrived. If the immune system reacts on the first dose, it is not reacting to the drug. It is reacting to something it already knows, something it was already armed against. His team builds a custom IgE assay, a blood test to detect antibodies specifically targeted against cetuximab. What they find: every patient who reacted had high levels of pre-existing IgE. They came to the infusion center already sensitized. The drug didn't cause the reaction. The drug was just the delivery vehicle for something the immune system had been hunting for years. Cetuximab is a chimeric monoclonal antibody, part mouse, part human. Its antigen-binding arms come from mouse antibody genes. Its scaffold is human. It is produced in living mouse cells, cultured in bioreactors. And here is where the story turns biochemical and fascinating. Proteins made in living cells are rarely delivered bare. They are decorated, a process called post-translational modification. One of the most important is glycosylation. These sugar chains are not random. They are built by a set of enzymes in the cell's Golgi apparatus, a kind of molecular finishing factory that adds sugars in a precise sequence. Mouse cells have an enzyme that human cells do not. This enzyme attaches a galactose sugar in a very specific orientation. The result is a sugar tag called alpha-gal. The drug was not the allergen. The sugar tag on the drug was the allergen, and the patients had been primed against that sugar long before cetuximab existed. Virtually every mammal on Earth, except humans, apes, and old-world monkeys, is covered in alpha-gal. Our primate ancestors lost the ability to make it approximately 28 million years ago. A mutation silenced the gene. Why? The leading hypothesis is that alpha-gal was being exploited by ancient pathogens, possibly the predecessors of malaria, as a surface docking site. Evolution kept the mutation. The consequence: our immune systems have never been tolerized to alpha-gal. We don't produce it, so our bodies never learn to ignore it. Every non-primate mammal's tissue is saturated with alpha-gal. When you eat beef, pork, or lamb, you are consuming the muscle cells of animals whose surfaces are covered in it. Glycolipids are lipid molecules that form the structural fabric of cell membranes. In mammalian meat, these alpha-gal tags are particularly abundant in fat-rich tissues. When a sensitized person eats this meat, those alpha-gal-bearing glycolipids are not absorbed quickly. They require enzymatic digestion. This takes time, 2 to 6 hours, which is precisely why the reactions are so terrifyingly delayed. The team now had the molecule, but they still didn't know how patients had become sensitized. The answer was hiding in geography. The worst hit centers were all in the same region. Someone on the team pulled up a CDC map of Rocky Mountain spotted fever distribution. The overlap was almost perfect. RMSF and the cetuximab reactions were sharing a zip code. They share a zip code because both cluster where the Lone Star tick clusters. The tick was the connection. We just hadn't asked our patients about ticks yet. Scott Cummins called back every patient in their study, 130 people, and asked a question nobody had thought to ask. Have you been bitten by ticks? More than 90% said yes. The suspect is identified. But how does a tick create an antibody against a sugar? The answer goes deep into immunology. Next time on the Midnight Allergen, the tick did it.